It is my great pleasure to welcome you to our inaugural Mary Kay Award ceremony. My name is Sabine Eming and I'm Professor of Dermatology at the University of Cologne, and I'm current president of the ESDR. And I would like to give a special warm welcome to Dr. Lucy GANIER, Mary Kay Chief Innovation Officer. And on behalf of the entire ESDR board, I would actually like to thank you and Mary Kay for this new partnership with ESDR. And what we particularly appreciate is that actually with this award, you support one of the ESDR most important values and mission, which is to support the early career skin scientists. And here I would actually also like to take the opportunity to welcome and thank my colleagues, Professor Eli Sprecher from Tel Aviv, who's our president-elect of ESDR, Professor Kilian Eyrich from Freiburg, who represents the grant committee. Thank you very much for your work on this. And I would also like to give a special thanks to Caroline Blondel-Baldassare from the ESDR Business Office, who is our corporate relations manager. Thank you very much for the fine organization of this ceremony. And before I hand now over to Dr. GANIER to introduce our awardees, please, Eli and Kilian, take the word.
Yeah, thank you very much, dear Sabine. My name is Kilian Eyrich from the University of Freiburg, and I had the honor, together with 3 other members of the ESDR board, to review this year's Mary Kay ESDR Skin Health, Skin Disease Research Award applications. And we have received numerous applications. There were 28 applications in total. Big names that we all know very well. And and not only I, but the whole committee was really impressed by the high quality of the applications, which show the high prestigious grant that we get here. So I can only say that there were many grants that would have deserved a lot more money even than we can give here. It was a very high competition, and that means that the winners could not afford even one negative remark. So even more congratulations also from our side of the committee here. Thank you very much.
Yes, hi everybody. My name is Eli Sprecher. I'm the chair of the Department of Dermatology at the Tel Aviv Medical Center. And it's really an honor and a great pleasure for me to be here today. I think that, as Sabine mentioned, we are particularly grateful to the Mary Kay for the opportunity to implement one of our major missions, which is really to promote forward, nurture research in dermatology. And I'd like to very much thank Mary Kay Foundation for the opportunity to do this. And also I'd like to very much thank the review committee, who has a very hard work. So just to give you a short overview of what is going to happen right now, we will first have an introductory and congratulatory remarks by Lucy on behalf of Mary Kay. And then we will have 2 short presentations. At the end of those 2 presentations, we will have a Q&A session. So please forward your question to the chat or the Q&A within the Zoom, and Kilian and myself will, at the end of the lecture, be delighted to ask your questions and get our awardees' answers. So thank you very much, and enjoy. Lucy, to you.
Great, thank you so much. Well, as you can hear, we're very excited about kicking off this inaugural event. And thank you, Sabine, for the introduction. Thank you so much, Ellie, for your work on the committee. And thank you so much, Kilian, as well. So as Sabine mentioned, I'm Dr. Lucy Gilday, Chief Innovation Officer of Product and Science here at Mary Kay. And before we get into sharing with you the awardees and recipients, I'd like to give you a little bit of background about Mary Kay and our mission. We're a decades-long leader in the global beauty industry, and our mission remains committed to enriching women's lives around the world. And we achieve this mission through research-driven innovation to provide the highest quality products. And we are very steadfast in our commitment to skin science research. Our team of scientists are constantly studying skin biology to develop new technologies and innovations with the highest efficacy and safety standards for our customers. We're also a company that firmly believes that a collaborative effort across academia and the scientific community is absolutely crucial to identifying new discoveries and driving innovation. ESDR brings together the brightest minds working to advance the future of the field, which is why we are excited to award those making an impact in the field of skin health research. And I'm excited to be here today to announce 2 recipients of $20,000 grants for the inaugural Mary Kay European Society for Dermatological Research Skin Health Research Grants. Now, as you heard, these individuals were carefully chosen amongst a lot of submitters in collaboration with the ESDR Research Review Committee. And we are absolutely thrilled to support the new discoveries in skin health or skin disease that each of the recipients will uncover. So first, I'd like to introduce our awardee, Dr. Michael Cangkrama. He's a senior scientist in the Department of Biology at the Institute of Molecular Health Sciences at the Swiss Federal Institute of Technology in Zurich. A key area of interest for Dr. Cangkrama is an understanding the molecular and cellular mechanisms underlying the development and progression of epithelial skin cancers, one of the most common types of cancers in humans. His research will focus on investigating molecular mechanisms of squamous cancer development and the role of the tumor microenvironment using state-of-the-art approaches, including tumorigenesis studies and 3D skin equivalents, advanced imaging technologies, and multi-omics approaches to identify the molecular drivers of squamous cancers and novel targets for their treatment. In addition to his scientific skills, Dr. Cangkrama is also a teacher at the Swiss Federal Institute of Technology in Zurich. His career goal is to become an independent researcher in the field of molecular skin cancer research. The Mary Kay-ESDR Research Grant allows him to further develop his independent projects as he continues to pursue independent group leader or assistant professor positions in Europe. So Dr. Cangkrama, please share your research project.
Right. Thank you so much, Dr. Lucy Gilday, for the kind introduction. So I would like to share my screen at the moment. Hopefully you could see the screen. Okay. So thank you so much again for the kind introduction. And I'm truly honored to receive this award from Mary Kay and the European Society of Dermatological Research. Now, I can imagine some of you might be wondering, what are you looking at on this particular slide? Well, this is a skin fibroblast that has been exposed to cancer cells. For years, we have known that fibroblasts play a crucial role in maintaining healthy skin. They are responsible for producing collagen, which is what keeps our skin youthful. But there is a darker side to the skin fibroblasts as well. In some cases, this cell can promote the skin cancer formations such as squamous cell carcinoma or SCC and basal cell carcinoma or BCC. And what's even more intriguing is that we have recently discovered a new way in which skin fibroblasts can facilitate cancer cell invasion through a process called mitochondrial transfer. So get ready, because I'm about to take you on a journey into the fascinating world of fibroblasts and their role in skin cancer. Now, before we dive into how fibroblasts contribute to skin cancer through mitochondrial transfer, I want to draw your attention to the close relationship between fibroblasts and cancer cells. Through my research, I have focused on a protein called activin A, which play a vital role in communication between these two cell types. As you can see, cancer cells release activin A to turn normal skin fibroblasts into what we call the bad fibroblasts or cancer-associated fibroblasts. What's particularly important is that my work has shown that cancer-associated fibroblasts can release several factors that could promote cancer cell migration, proliferation, and even the survival. And I have identified the mechanism involved in these complex cell-cell interactions. But there's still so much we do not know about activin A's role in skin cancer. Right now, I am exploring the question whether activin A regulates other behaviors that promote tumor growth by skin fibroblasts. Now, I want to share with you another remarkable role of activin A in skin fibroblasts that I have recently discovered. It turns out when we treat skin fibroblasts with activin A, they form these unique structures called tunneling nanotubes, or TNTs for short. TNTs are essentially long thin tubes that form between cells and serve as a means of communication. So they can bridge cells together and allow the exchange of information, nutrients, or even organelles. Importantly, activin A is able to promote the formation of these TNTs. And here's where it gets really interesting. One emerging role of TNTs that they are used for the intracellular transfer of mitochondria. So mitochondrial transfer is a truly fascinating phenomenon that has been well established in the field of neurodegenerative diseases. In conditions like Alzheimer's and Parkinson's, healthy microglia have been found to transfer their healthy mitochondria to damaged cells, preventing them from dying. It's an amazing process, and it was beautifully presented in one of the papers published in Cell recently. But that's not all. Our own research has uncovered something equally remarkable. We have observed that a transfer of mitochondria from skin cancer cells to skin fibroblasts, and we were able to capture this process in this immunofluorescence image. Importantly, we can even capture the transfer of mitochondria in real time. In our experiments, we label the skin cancer in red and their mitochondria in green. So while skin fibroblasts were left unlabeled. As you can see in this video, within just 6 hours of co-culturing these cells together, we observe a delicate yet stunning transfer of these green labeled mitochondria from the cancer cells to fibroblasts. Now, the important question is what happened to these fibroblasts after receiving mitochondria from the cancer cells? To answer this, we isolated mitochondria directly from the cancer cells and transplanted them into skin fibroblasts using this technique called mitoception. The recipient fibroblasts, which we call mitocepted fibroblasts, are quite fascinating. Once they receive mitochondria from cancer cells, they become pro-tumorigenic cancer-associated fibroblasts. So they express high level of cancer-associated fibroblast markers such as activin A and the interleukins. They also proliferate much faster compared to the control fibroblasts. All are hallmarks of these pro-tumorigenic cancer-associated fibroblasts. But it doesn't stop there. When we collected the growth factors and the factors produced by these mitocepted fibroblasts and exposed them to cancer cells, we observed a significant increase in cancer cell proliferations. So this is a clear indication that the mitochondrial transfer play a critical role in cancer progression and eventually the invasion. Another important question is that, can we modulate mitochondrial transfer using an unbiased approach? For this purpose, we utilize state-of-the-art technologies to synthetically introduce mitochondria into fibroblasts using fluid FM. In our experiment, we extracted green label mitochondria from cancer cells and directly injected them into fibroblasts. As you can observe the insertion of this green label mitochondria in this video. Remarkably, after we injected these mitochondria into these fibroblasts became more proliferative, behaving like a pro-tumorigenic cancer-associated fibroblasts. So taken all together, in conclusion, the mitochondria exchange in the tumor microenvironment is a new and exciting field in cancer research, particularly in skin cancer, where it remains largely unexplored. Today, I have shown how cancer cells can transfer mitochondria to neighboring fibroblasts, turning them into pro-tumorigenic cancer-associated fibroblasts, and eventually promoting cancer cell survival. So the question, what's next? With the generous support from Mary Kay and ESDR, I will continue my research to explore the most important aspect of my work. This includes investigating the mechanism that regulates the mitochondrial transfer in this vicious cycle between cancer cells and the skin fibroblasts, as well as exploring ways to inhibit this process as a potential therapeutic target for skin cancer treatment. For example, using a well-established 3D organotypic skin culture. So I'm excited to continue my research with the support and share my progress with you in the near future. So this leads me to the most important slide of my presentation today, the acknowledgement. So on this final slide, I would like to express my gratitude, especially to my great mentor, Professor Sabina Werner, whose constant support has made this award and my research possible. I also want to thank all my colleagues and collaborators who have contributed to this work. And importantly, I want to give a big shout out to my family and friends who are watching this probably from Indonesia, Australia, and also in Switzerland. Thank you all so much for your support and for your kind attention. Thank you.
Thank you, Dr. Cangkrama. That was very exciting. And I'm going to hold my questions for the Q&A session at the end, but we're really looking forward to seeing your progression in the mechanism of action work that this research grant is going to help fund. So thank you very much and congratulations. So next, I'd like to introduce the second awardee for the Mary Kay-ESDR Skin Research Grant. And this is Dr. Clarisse GANIER. She's a postdoctoral research associate in the Center for Gene Therapy and Regenerative Medicine at King's College London in the United Kingdom. Her work, along with others, is focused on the Human Cell Atlas Project, which is an international collaborative effort to map all the cell types within the human body to generate a comprehensive skin cell atlas. As part of this work, they've created a multi-scale spatial atlas of healthy skin and basal cell carcinoma incorporating single-cell transcriptomics, in vivo optical coherence tomography, spatial global transcriptional profiling, and in situ sequencing. Among many community engagement activities, Clarisse co-founded and currently leads an association called the London Postdoc Network, which aims to provide access to career development opportunities to postdocs. The Mary Kay-ESDR Research Grant allows her to continue to pursue her professional goals. And in the future, she would like to start her own laboratory focusing on skin cancer biology by combining multiomics data in vitro and ex vivo human skin cancer models. So I'd like to ask Dr. GANIER to share a short presentation on her research.
Thank you very much, Dr. Clarisse GANIER, for this really kind introduction. So I'm going to share my screen right now. And yeah, I want to thanks as well, everyone, for joining today. It's really my great pleasure to be here and to present this project that has been generously funded by Mary Kay-ESDR. So this project aims at deciphering the roles and interaction of keratin-17 tumor cells and the proximal periostin fibroblasts in tumor progression and invasion. So to answer this important question, the model is going to be non-melanoma skin cancer. So the two main non-melanoma skin cancer are the basal cell carcinoma BCC and squamous cell carcinoma SCC. They are the most frequent malignancy in human and represent a major cost to the health system. So BCC is about 67% of those non-melanoma skin cancer and SCC 33% based on the US numbers. And those number doesn't stop to increase since here. So it is really important to understand better this cancer, the interaction between the cancer cells and the tumor microenvironment, the fibroblast. As my colleague also really well demonstrated, it is a major player in the cancer progression. So in this project here, by understanding this interaction, we aim to find therapeutic targets. So in a previous work that aims to answer the question of how the skin is organized at different anatomical sites in normal body and normal face, and also in a particular cancer, basal cell carcinoma, we have sequenced cells from ELFI and BCC using single-cell RNA sequencing, spatial transcriptomic, the 10x Visium method, and in situ sequencing. And we have done also some in vivo optical current tomography imaging on humans. This nice technology of spatial transcriptomics, so the first one we use at 50 micron resolution, allowed us to find specific characteristic and map the cell types in the ELFI face, ELFI body, and in BCC. In order to complement this technique with a single cell resolution, we did an in situ sequencing that allows us to have a custom panel of genes designed to find all the skin cell types. So we have here global transcriptional profiling at 50 micron resolution and a complementary method of in situ sequencing at single cell resolution with specific genes for skin cell types. Those methods allowed us, so here to just really show you the single cell resolution and those nice transcripts. So as I said, those methods allowed us to find that keratin-17 was really upregulated in tumor cells of basal cell carcinoma. So this we could find in single cell RNA sequencing, in spatial transcriptomic, in the global transcript profiling, but as well, if we look really specifically into the section, we could find that this upregulation was really in tumors areas. This proteins is also found in healthy condition in the hair follicles area, as you can see here, and also we can see here and here in this immunostaining. But it was particularly expressed in the cancers area, as you can see in situ sequencing and immunostaining here. So we know that keratin-17 promotes skin cancer cell progression, migration, and metastatic potential in many other cancers, so breast cancer, pancreatic cancer, and also lung cancer. And we know that this upregulation is also in skin, non-melanoma skin cancer. But the role remains unclear, and the interaction with the cancer stroma, in particular the proximal cancer-associated fibroblasts, has not been explored yet. And we know that as in healthy skin, the cancer-associated fibroblasts are really heterogeneous. So in this project, we could nicely in healthy skin characterize the heterogeneity in this fibroblast. So we find mainly 4 main populations of fibroblasts. And the one that is in particular interest is this population F3 here, that is mainly in colocalization with the hair follicles in healthy, So that also is in close colloquialization with this keratin 17 expression in healthy. And this particular population is the one that is really well expanded in the BCC and in close proximity with the tumor islands and the invasive area of the cancer. So this particular subpopulation express mainly periostin, post-TN. So we could find this finding in the 10x Visium, but also in the in situ sequencing dose. So that was kind of a robust result. So the aim here now is to look specifically at those periostin fibroblasts in non-melanoma skin cancer. So this that we have found in BCC, we wanted to confirm that it was the case in SCC. So this is actually some preliminary result of this project. And we could nicely see this upregulation of keratin 17 in the SCC within the SCC area, lack in BCC, and also this periostin post-TN expression in the stroma around the SCC, in SCC, like in BCC. And we have confirmed this method directly on the section from the SCC and BCC biopsies with the immunostaining of keratin 17 nicely organized in BCC, and this post-TN in red dots of the RNA around it. And this staining was actually not as structured, but this is the nature of this cancer, the SCC, within the cancer cells. And we could nicely see this post-TN RNA expression in the stroma as well, and also within the cancer. So now we want to understand what are the roles and interaction of those keratin 17 tumor cells and proximal posterior cancer-associated fibroblasts in tumor progression and invasion of non-melanoma skin cancer. So the plan of that project is to first establish a 3D in vitro non-melanoma skin model using decellularized human dermis. So we did already isolated in the lab BCC fibroblast keratinocytes from BCC, and we are doing the same with SCC. And we want to embed them into this decellularized human dermis to see if they can replicate those invasive and nodular properties. The second aim is to engineer knockout keratin 17 BCC and SCC tumor cells and knock-in of keratin 17 in healthy primary keratinocyte using CRISPR-Cas9 technology to analyze the invasive properties of keratin 17 knockout and knock-in embedded in the 3D model that will contain either healthy fibroblast or cancer-associated fibroblast. And the third aim is to identify and validate new tumor-promoting signaling pathways in 3D models of non-melanoma skin cancer. And for that, we're going to do in silico interactome analysis of our previously generated data set for BCC, take some publicly available data set for SCC, and we already have the spatial transcriptomic for both. And we want to validate these newfound interactors and key signaling pathway genes in our in vitro 3D models. And so for this, I would like to mainly thank Jay Roy, who is a PhD student working with me on this particular project, my boss and mentor, Professor Fiona Watt, my second boss, Magnus Lynch, who is really helpful of providing the skin biopsies, all the wonderful Watt Lab people and Hanifa's Lab people who are joined really recently as well, the Human Cell Atlas Consortium for allowing me to gather all this data that lead me to this project today, the patient for the skin biopsy, which we couldn't do anything if they were not generously giving it to us, and of course, the Mary Kay-ESDR grants for giving me this opportunity to do this project and trust me on this. Big thank you for those two funders. And I would like to give a special thanks actually to the ESDR that was always supportive of early career researcher through grants, research grants, travel grants, and also through great trainings. Thank you so much for the support, and thank you everyone for listening to me today.
Thank you very much, Clarisse and Michael. These were really outstanding talks. Let's see if we have any questions. So we have one question regarding the CF. It's a question to you, Michael. Will blocking of activin A be a more efficient way to block the formation of CAFs?
But that's a really interesting question because in a tumor microenvironment, there's a heterogeneity of the fibroblast populations. And we found out that inhibiting activin A, for example, using the antagonist follistatin, could inhibit certain phenotype of cancer-associated fibroblasts, for example, cell migration, proliferations. But back to the heterogeneity again, there's so many signaling that are controlling this formation of cancer-associated fibroblasts. And we believe that activin signaling, by blocking them, could actually also inhibit formation of these cancer-associated fibroblasts.
Thank you very much. Kilian.
Yeah, also from my side, I think we've really chose the right candidates. Congratulations. Clarisse, I would have a very short question to you. The two markers that you mentioned, keratin 17 and periostin. Keratin 17 is associated to be an autoantigen in psoriasis, while periostin is rather type 2 immunity associated and a response marker for example, interleukin-13 therapies. So one is rather psoriasis like Th17, the other Th2. In that context, do you analyze the immune cell infiltrate also in these tumors?
So that's a really good question. We actually started a collaborative project with Ms. Hanifa. So that's how I kind of joined a lab as a visiting researcher to study the immune components and cellular mechanism behind it in BCC based on all the data set that has been generated already. But so far, we focused really on epithelial and mesenchymal compartment of LV and BCC. So I'm going to have an answer to you, I think, in a couple of months. But there is a big change, and there is mainly a change in the T cells that show actually a separate cluster that appears only in BCC and in macrophages, in relationship to fibroblasts, actually, to the CAF that are around. But the question still remains, which kind of CAF? Because as I said, this project is going to focus particularly on the proximal CAF, but the distant actually are the ones that play the bigger role in the immunity.
Right, thank you. And Sandrine, as always, had a very similar question, just more elegantly formulated. But I think for reasons of timing, Eli, we need to proceed here. We could ask many more questions. Thanks a lot, you two.
Thank you so much.
Thank you very much. And now we will have final words by Sabine and Lucy. Please.
Good. So congratulations also from my side to both of you, Michael and Clarisse. So that was very interesting, and I wish you all the luck for your experiments, and I'm very curious to follow up on those. So thank you very much to everybody who contributed to this, and special thanks to Mary Kay, who made this possible. Dr. GANIER, you have the final word.
Thank you, Sabine. And again, as everyone has said here today, it's been great to actually kick off the inaugural partnership between Mary Kay and ESDR for these research grants. And I've just been very excited to meet and see the presentations by Clarisse and Michael. And so I want to congratulate both of you as the grant award recipients. And you know, what you can hear is we just can't wait to see what's next. We're all excited, so we look forward to hearing updates, but also we just can't wait to see the impactful contributions you actually bring to the future of skin health, right? And the considered research. You know, it's been quite some time since I was in graduate school and in postdoc, and so I just really, really love seeing our young scientists really dedicated, and it's so great to actually partner there. So I want to congratulate you, but thank all of the applicants who submitted proposals. And Sabine, to you and our ESDR partners, you know, for your collaboration throughout this process. It is the first year, and I'm very excited about future connections with ESDR. So I want to thank everyone and actually wish you all to enjoy the inaugural International Societies for Investigative Dermatology Conference, which is actually coming up in the next coming week. So everyone, thank you for your participation today and have a great week.
Thank you so much. Bye-bye.
Bye-bye.
Thank you very much. Bye.
Thank you.